the countdown. But I've started the recording. I'm gonna actually go on mute now, and then you can start essentially whenever you're ready.
Okay, sounds great. Thanks. Okay, hi everyone. I'm really excited to tell you today of some of the research in my lab. We're going to talk about what the body can actually tell us about the brain and how in our lab we study machine learning methods for automated quantification of behavior. Just a bit about myself. I'm a PI at the Tsai Institute here at Yale University. I'm in the Department of Biomedical Engineering. I have a lab that's fairly new, and I'm really excited to share some of our recent research, as well as, please talk to me offline if you have any questions. I'd be really excited to talk. Okay, so we're going to get started today with a video, actually. In this case, we see a pack of wolves chasing a hare, per hare, but really consider the task for just one wolf in the pack. It needs to process the visual signal of this hare, understand that it's prey, and output the right muscle signals to try to track the hare. And what about these two wolves? They have the same goal, right? They're chasing the hare, but are they doing it in the same way? And finally, here the wolves decide to work together. So any one wolf needs to interact socially and cooperate with others in the pack to be able to achieve this common goal. Here they do achieve it. The hare does not survive, poor hare. Really, there was a pack of wolves chasing it, and that's super surprising. But really what we want to figure out, or what we study, is the task that any one wolf performs, which is very complex, and we really want to figure out its neural underpinnings. Okay, very complex, right? As a field, we're actually becoming very adept at recording from a large number of neurons simultaneously. Of course, we don't usually record from wolves, but still, what can we actually do with neural data that's recorded from the brain? We could learn machine learning models on it, and these models have become excellent at modeling high-dimensional data. But while artificial neural networks and other machine learning models work very well in the data regime that they were trained, they don't always extrapolate well, and they don't always lead to insight about behavior or even sometimes neural insight. So what can we do to help? We could actually integrate these models with our understanding of behavior. We actually know a lot about behavior. We have some knowledge of biophysics in the behavior, and also some knowledge of anatomical constraints, both in behavior and neural activity. So today I'll show you how this knowledge, these two pieces, biophysics and anatomy, can actually lead to more generalizable and interpretable models of not just behavior, but also neural insights. Great, so let's get started. Well, machine learning models and melding them with data, biophysics, and anatomy is exactly what I focus on in my lab. And here we study complex systems, and these three pieces are very key or integral to constraining our models to be able to extrapolate and draw insights into behavioral neural functioning. So I will today talk about three different projects that really focus on how behavior can really provide a window into the brain. Great, so let's get started with our first goal. I want you first to consider what happens as we're learning a new movement. Let's say we're learning a new salsa dance move. Well, it doesn't take some of us too long, not me, but some of us can learn it very fast, even though the muscle activity that is created while performing this new movement needs to be drastically different from other movements that we're more familiar with, right? But the relationship from the muscles here, from the muscles in blue to the actual kinematics stays the same, right? So if the brain actually knows about these relationships or these constraints in order to achieve generalization, it is possible that we can also learn a generalizable model of how neural activity leads to movements. So in this part of the talk, I'm really going to talk about incorporating biomechanical constraints from behavior to improve generalization for neural models. But I want you to first take a simpler case about generalization or kind of achieving new movements. I want you to consider the case of cycling slowly versus cycling fast. When you're cycling fast, not only are the muscles that you're contracting, not only are these muscles contracting faster, some may also have higher amplitudes and completely different patterns of contraction. However, once you actually learn a movement at one speed, we want to be able to generalize it to another speed, right? If you're tracking something that's moving continuously, you want to be able to track it at any speed possible. So there needs to be somewhere in the brain that takes in feedback about what you're tracking and can have a continuous representation of how that target movement maps into varied muscle activity and how that is translated into actual movement. So is this in the motor cortex, and how exactly does the motor cortex do it? Well, I'm going to tell you briefly about the experiments that were done in order to answer these questions. Experiments here were done by Abigail Russo in Mark Churchill's lab at Columbia University, where they trained 2 monkeys to cycle through a virtual environment while tracking a target continuously. This target itself was moving at different speeds. And as you can see, the monkey's speed also changes continuously in order to keep on top of it, in order to stay on top of it. Now, if we actually take a look at the neural data that was recorded at the same time during these experiments, and these are single unit neural recordings from the motor cortex, muscle activity was also recorded, but let's take a look at the motor cortex activity first. So what does the motor cortex activity look like here? I'm showing 3 different neurons, and it's firing rates from the slowest to the fastest movements. And really, here you see very different patterns in the neural activity. Sometimes the neuron has a higher amplitude during the slower movements, sometimes during the faster ones. Now, what we wanted to ask was, how can we understand this neural activity? And how can we understand it using, for example, behavior? Now, what does understanding mean? I take it as prediction. Can we actually predict neural activity using the behavior that was recorded? It turns out that kinematic activity in the behavior doesn't actually lead to a good prediction of the motor cortex activity. It doesn't decode it very well. Similarly, we have the activity of all the muscles that were also recorded, and I'm displaying 3 of them here, but they don't actually do a very good job of explaining that neural activity either, as I'll show you later. Now, although this didn't hold true in this data set, this hypothesis that we can actually predict neural activity from behavior is potentially very useful because it can give us clear quantitative predictions that we can make about the neural activity with changes in behavioral conditions. So how else can we get clear predictions about neural activity given behavior? Well, one way to get predictions is to consider a goal-driven approach. Here we have the same muscles that we were plotting earlier, now in state space using principal component analysis in order to actually see what the structure looks like in the muscle activity. Now we can consider the goal of the motor cortex as generating the right muscle activity to produce movements. So we can ask ourselves, what would a neural network do in order to actually produce this muscle activity? Well, we actually just, to find out, we trained a neural network. We trained here a recurrent neural network to model the function of the motor cortex. So here we trained it such that it has the same inputs and outputs as the motor cortex does. So here with speed as an input, as a hypothesized input to the motor cortex, and the muscle activity as an output. Now I'll be referring to this as the open loop recurrent neural network, which should give you an idea about what's coming next. But here, just to continue, once it's trained, we can plot the activity of the network's nodes in principal component space here for each speed. And this activity seems to follow a very clear structure, which actually looks very different from the muscle activity structure. So there's more going on in the neural space than just representation of muscle activity. Now, why is this useful? This network is actually useful because we can decode the neural signals better using this RNN, the recurrent neural network activity, than if we had just considered the muscle data. So here I'm showing you an example neuron's firing rate in white and the corresponding fit using the recurrent neural network in green as compared to the corresponding fit using the muscle activity in brown. And you can see that the RNN here fits this neuron much better than the muscle activity. And it's not just true for this one neuron. In fact, single neurons as a whole are fit much better using the RNN as compared to the muscle activity. So the RNN here is a good model to consider for the motor cortex, right? And here, like I said, we just use the behavior in order to constrain our model. But is this the entire picture? I want to actually consider what the goal of the motor pathways is in the brain. how can we best emulate the task that the motor cortex actually performs? So to understand this better, we have to zoom out a little. In fact, the motor cortex does not actually work in a silo, right? It receives constant feedback, including visual and proprioceptive. And the end movement is always achieved using the musculoskeletal system, which importantly has its own dynamics. It's a physical system with inertia and its own biomechanics. So the motor pathways can really be thought of as a controller for the musculoskeletal system in order to achieve a desired movement. Now, does this viewpoint actually help us understand the neuron's activity better? Does modeling the musculoskeletal system actually help us? Well, in fact, this idea has been kind of around for a little while now. It's also termed optimal feedback control in the motor control literature. And it's a straightforward formulation in an idealized setting where the muscles are linear and the controller is optimal. But the issues are that the muscles are not informed by anatomy in the past studies. And the controller is not informed by neural data. Why is this important? It's important because in this case, for example, in the case of diseases and disorders, we may actually like to design appropriate neurostimulation, but we don't know how to interact with the system if we don't know exactly where the computations are taking place. So we really need to understand not just what the goal of these regions is, but where exactly and how exactly these computations are taking place in order to interface with the system. So to better model this end-to-end process, we have to build in the details of the musculoskeletal system. So can we understand more about the brain by actually considering the neural activity as a controller for the musculoskeletal system? This is exactly what one of the grad students in my lab, Noman Ilvani, is working on. And he did exactly this. He developed first a fairly detailed musculoskeletal model of the monkey in a software called Mojoko, which is a physics-based simulator that really models the muscle activity as well as musculoskeletal system dynamics very well. Now, for learning a model to actually drive this non-human primate arm, essentially, we're driving the muscles that lead to kinematic output. So we need a controller providing a high-dimensional signal to this arm model. Now we know that a recurrent neural network structure works very well and has been used in the past to model a motor cortex. So that's exactly where we start. This RNN directly takes in feedback provided by the arm model, including proprioceptive feedback and the target or goal information, and it's really set up to output the muscle activations. Now, Noman trains this all using deep reinforcement learning in a soft actor critic setup to train this RNN actor network, as he's calling it, with a reward function to track experimentally recorded kinematics. Now, when trained, we have a model that achieves recorded monkey behavior, all without using the neural recordings during training. That's the beauty of it. It's really just using behavior to get predictions. So we started actually by applying this model to this arm, the monkey arm that we are looking at now. And we trained it for the cycling task that we saw earlier with the non-human primate performing cycling. And it really works, right? We're seeing very clear cycling movement. The musculoskeletal arm actually produces experimentally recorded kinematics, as you can see in this video, with high accuracy. And for reference, this is the monkey performing the same task. Now, I know, this may not seem impressive, but the arm has to learn from scratch in order to actually get to this stage. So through this method, we have a network that can actually output accurate kinematics in conjunction with an anatomically accurate musculoskeletal model. And, we trained it to do so it's not particularly surprising that it can actually reproduce the kinematics. But we can't even think about, for example, perturbations in an open loop setting. And here, we can actually look at what perturbations in the muscle activity or in the kinematics, what these perturbations actually result in the neural space or in the network space. And these perturbations can be corrected very well with our neural networks. So that's something that's really nice to see. And again, can't really think of even performing this kind of a study in an open loop setting. So that's really an advantage of using closed loop models. Now, how does this do when looking at corresponding fit to neural activity? That's what we were also interested in, right? How can we best use and model the behavior in order to tell us something about neurons? So we can actually fit models, here a linear regressor from the neural network's activity to each neuron's activity. So here on the y-axis, we are showing kind of fits to each neuron's firing rate, and the y-axis is the r squared, a fit to that neuron's firing rate. And on the x-axis is if we had actually predicted that neuron's activity using just the kinematics or just the muscle activity. And if our dot here, so each dot here is a neuron, and if our dots lie above the dashed y equals x line, that means that we are doing better than the corresponding other comparison models on the x-axis. And as you can see here, we are doing better for most of the neurons to actually predict the activity of the neurons using our networks. Now, this is great because again, we hadn't used neural activity while actually making our models, while creating our models. But what about generalization? That's actually what we had also started off being interested in. And here we see that these networks actually generalize to single neuron firing rates in unseen conditions. That's been a very promising avenue, because generalization is typically very difficult to do in actual neural networks. So here we see on the x-axis, we're comparing to an open loop RNN, which is basically what we started out looking at. So this open loop RNN does not have any idea of the biomechanical constraints or the biophysics of the actual behavior. It just is outputting muscle activations. So it doesn't have any feedback. It doesn't actually know anything about what the body is doing. And if we compare our models to the open loop model on conditions that are basically an interpolation of previous conditions, we are already doing much better than on the y-axis. We see that we're doing much better than the comparison open loop model, because almost every dot is above the y equals x dashed line. So the neurons are predicted much better using our models, but where we really excel is actually extrapolation. So if we now compare the fits to the predictions to neural activity in conditions that are very far away from what the models were trained on, we are still doing really well in predicting their activity. And we think that that's because we are actually building in more information in our models about behavior, about how behavior in all its complexity, in all its glory, in all its non-linearity. So this R model, for example, is a highly nonlinear dynamical model that we are really taking into account. We're really modeling it very well. And that is what's allowing our models to actually extrapolate in their prediction of neural activity that is actually driving this behavior. So this is a very exciting kind of line of work for us. And the generalization that you just saw to unseen conditions is really, really, really excellent. But we also wanted to see if we actually have access to not just the behavior, but also some of the neurons' activity. Could we also, how does that help in terms of our generalization to unseen conditions? And in fact, if we constrain some of our neural network nodes to known neural activity, we can actually extrapolate even better. So this is really now a data and goal-driven network, and we really see that the generalization capabilities are quite extraordinary. So in this part of the talk, I really focused on how the biophysics of the body can help us understand something about the motor pathways that are driving this biophysics, that are driving the body itself. Now, what we're really excited about is continuing this work with the help of the Brain Initiative, as well as looking at multitask generalization in these models. To really see if we actually try to train our models on one task and then try to test on another. We are actually seeing that we can actually perform much better in terms of understanding the neural basis of multitask generalization. But I really encourage all of you to go check out the paper that we just put up on bioRxiv. It's the last link in the list of references. Okay, so this is the first part of the talk that I was excited to present to all of you. Next, I wanted to address our take on modeling the activity of multiple subjects performing the same task. How do we actually identify the similarities and differences in the way that they perform the task? Now, I'm going to ask you to think about the wolves again. There were two wolves that were actually performing the same task. They're trying to catch the hare. Are they doing it similarly? Are they doing it differently? And really, here we're basically asking a simple question: if two subjects are performing the same task, can we understand the similarities both in the neural activity as well as the behavior during these tasks? Okay, so this question really is only possible to examine at the neural level due to significant advances in recording technologies like neuropixels and wide field calcium imaging, which have really led to an explosion in the number of neurons that we can record simultaneously, at least from the mouse brain and upcoming in the non-human primate brain as well. And in a review article that's listed here, I really talk about the benefits of population analyses for neural data. But what about the behavior? Can we even pick out, why do we really want to pick out similarities in behavior here? It's because the mice are performing this task in potentially slightly different ways. So they're performing many different kinds of movements during even a simple task. Here it's a two alternate first choice task that the mice are performing, but there are many different kinds of movements. For example, sometimes they just stop doing the task and start grooming. But can we actually pick out where in this task, in an automated way, can we pick out where these mice are performing spontaneous, uninstructed movements as compared to really the task-driven movements like licking the spout and pulling the lever? Just as an aside, this data was recorded in Anderson's laboratory at Cold Spring Harbor at the time by Simon Musil, and this is the data that I'll be talking about for this part of the talk. So we don't actually have the tools yet to understand when in this behavior we see spontaneous movements as compared to task-aligned movements. But we do see repeated behaviors across these mice. So how can we begin to model these similarities? Well, one option is just to perform dimensionality reduction of these behaviors in order to understand where these behaviors are similar or different. But first, I want to just show you that naive dimensionality reduction methods don't actually work very well for this kind of high dimensional across subject data. In fact, there are many surface level differences between these mice that end up dominating the latent space. So here I'm showing you an autoencoder structure with a bottleneck layer to actually represent the latent variables. But if you try to model the activity of 4 different mice, for example, using this kind of a structure, and you want to pick out the points in time where mouse 1 and mouse 4, for example, are performing the same kind of behavior, for example, lever pull, it's very difficult to do so. But this nonlinear autoencoder approach does actually reconstruct the videos very efficiently. So we just need to add some interpretability in the latent space in order to recover where in time these behaviors are similar to each other as compared to different. And so that's exactly what another graduate student in the lab is working on, Dayao Yi. So she's expanding on this convolutional autoencoder structure to directly partition the subspace, the latent subspace, into different parts, one that's a cross subject and one that's subject specific. So she came up with a novel way to do this. She added constraints to our latents. In this case, we have some latent variables that produce the tracked body parts, for example, and some that are regularized to using the KL divergence to a standard normal distribution, and thus promotes similarities in the latent space, and others that actually promote separation in a constrained latent space using regularization to a pre-specified prior distribution using the Cauchy-Schwarz regularizer. Now, what does all that do? Well, in fact, it separates our latent space very cleanly into a cross subject at the top here, that is latents that are very similar across all these subjects, and latents that are subject specific at the bottom. That is latents that are just very separated or different across all the different subjects. What can that actually help us with? Well, one fun thing that we can actually do is just change the subject specific latents from one to the other. So here I'm changing one mouse latent in blue to another in red or green or orange. And in fact, what this does is, if changing this in the latent space, what this does is actually changes just the appearance of the mouse in this case, but really retains the actual task or actual behavior that they're doing here. They're licking the spout. All these mice are actually, all these images that are generated at the bottom, they're all licking the spout, but their appearance now is very different. So we can play fun little games like this. But really, our initial goal, was to really figure out what are the similarities in the mice that are actually performing this task. Are they doing it in the same way? Now we're calling this autoencoder structure a constrained subspace variational autoencoder, and this actually allows us to uncover common behavioral motifs across subjects in a completely unsupervised way. So for example, here you see that there are 3 different dogs, right, that are all barking. We can tell that as a human being, we can actually tell that they're all actually just barking. It's just the same behavioral motif that they're performing. We're calling this a motif. Which basically is a set of actions that are carried out by an animal over time, set of repeatable actions, and these set of actions, when put together, they form an ethogram that basically represents the entire behavior of the animal. Now, as I said, these kinds of motifs are very straightforward to tell by the eye, but it's actually difficult to recover the same behavioral motif across animals using machine learning algorithms. But here, we're using our constrained subspace variation autoencoder that actually is constrained in very specific ways. We can do that. So here I'm showing you how our model recovers across subject behavioral motifs in an unsupervised way. And we really see, for example, these specific behavioral motifs like licking or grooming being represented in the ethogram that I'm showing you at the top here. And we also see, in fact, that these kinds of more spontaneous movements like grooming occur at quite a variable frequency across animals. For example, mouse 1 and mouse 4 here are grooming much more often than mouse 2 and mouse 3. And we can recover this again in a completely automatic way. So that's been also a very nice study to do. And it allows us to ask questions about the neural activity and the neural basis for these behaviors very easily now. So for example, we can actually tell that the mice, when performing a lever pull, different mice have a very correlated neural activity across these brain regions that I'm plotting on the x-axis, and the neural correlations between subjects of what I'm plotting on the y-axis here. And we see that if we compare the correlations across mice, it's very comparable to the correlations in the same animal performing this task in different across different instances. And that's not true. While that's true, while these mice are performing the actual task like a lever pull, very specific task-related action, that's not true when the mice are just idle. So they can just be thinking about very different things. However, for grooming, for spontaneous, spontaneous action like grooming, we weren't sure what to expect. Would the neural correlations be similar across mice? And in fact, they do seem to be very similar across mice as they're performing this task, the grooming action. This is now very easy to do now that we have understood the behavior and automatically quantified it in a nice way. So here in this part of the talk, we were really modeling intersubject similarity and variability, actually. So now we're actually looking at how to quantify the variability across animals. Does everyone do the same task in the same way? Or are there many differences in how we do it? And what does that actually tell us about the neural activity as they are doing it? Okay, I know I'm really short on time, but I'll just end by saying that we're trying to also, we are really getting interested in modeling naturalistic behavior through all these different automated and machine learning approaches. And really the idea is to move from a very constrained lab setting to much more like the video that I showed you in the wild, and also considering social behavior interactions amongst animals in order to really understand behavior and neural activity in all its glory. So I'm going to skip through a couple of the slides here, but I'm really excited to model not just spontaneous activity, but also social behavior in collaboration with Nancy Filigrano, who you will also hear from in one of the other talks, as well as Dan Lesson at the University of Florida. And here we're really considering high dimensional activity or behavior that is really being modeled in latent spaces that are well constrained in order to really be able to have interpretable latent variables and inform us about not just the actual behavior, but also hierarchies in social behavior and spontaneous behavior. And we're really excited about continuing on this work using techniques in dynamical hierarchical clustering, as well as semi-supervised approaches to modeling behavior. So if you're really interested in that, please email me and talk to me offline. I'd be very happy to talk to you. And the questions we're addressing here are really to understand our, social, pro-social events in mice, as well as antisocial behaviors in mice, and the neural underpinnings of these behaviors. Okay, so I'll just end by reminding you all that I study complex systems in my lab. These three pieces, data, biophysics, and anatomy, and knowledge about the actual task, are really integral to constraining our models to be able to extrapolate and draw insights into behavior as well as neural functioning. And I just wanted to thank all the collaborators, as well as the funding sources, and the students in my lab, who are the ones who really focused on the actual research. And on the right here is the Tsai Institute building, which at Yale, which again I'd be very happy to talk about offline. And with that, I really want to thank all of you for listening, and please reach out if you have any questions. Thank you.